My name is Noha. I'll be presenting karyotypes and chromosome aberrations. What is a karyotype? What is a karyotype used for, and why is it important? A karyotype is the visual representation of the number and appearance of chromosomes in the nucleus of a eukaryotic cell. It's used as a laboratory test to examine a sample of chromosomes in a cell. It's to analyze those chromosomes to look for any major chromosomal aberration or anomaly. And this anomaly can be in the number of chromosomes, the banding pattern of the chromosomes, the shape, the number, or even the pairing of the chromosomes. So a karyotype can also help identify a genetic disorder as the cause of the disorder or disease. It can help with patient diagnosis of disease risk. Or maybe if a patient is aware of specific genetic risks that he or she may have, or gene mutation risks. Maybe if they're aware of this, they can have lifestyle changes that would prevent the expression of that gene or that mutation. Again, chromosomal number, banding, size, shape can also be seen in the karyotype. So karyotypes are very useful in medicine and for patient prevention. And they can even help with pharmacogenomics. So maybe if specific people have genes that will not interact well with specific drugs and will have worse side effects than other people, a karyotype can help in identifying those individuals so that they can take a different drug and have personalized medicine. So how do we perform a karyotype? So you can have a sample of blood or a tissue sample. So let's say we have a sample of blood that's been drawn. That would be our first step. The next step would be the mononuclear cells have to be purified from the blood by centrifugation through a dense medium. This allows the red blood cells and granulocytes to pellet, but holds the lymphocytes and monocytes. Next, we would culture or incubate this in the presence of a mitogen like PHA, and PHA stimulates rapid lymphocyte proliferation. Next, after the culture period, now we have a large population of dividing cells. And of course, we want our cells to be in prophase or metaphase because this is where we can most see our chromosomes, because this is when the chromosomes are condensed. So we use a drug that will disrupt the mitotic spindle and prevent the complete mitosis of the cells. So now this greatly enriches the population of the metaphase cells. Next, we treat with a hypotonic solution and then fixate the cells so that we can put them on a slide and we stain them so that we can see them, visualize them under the microscope. If it was heterochromatin, it would be darkly stained because it's tightly packed. If it's euchromatin, it would be lighter because it's lightly packed. And then now here we have a karyotype that we can use for visualization of genetic disorders, gene mutations, or chromosomal aberrations. So scientists use 3 key features to identify chromosomes. This would be number 1, the size of a chromosome. This is the easiest way to tell chromosomes apart, just by looking at their size. Next, we have the banding pattern. This, again, can help us with euchromatin and heterochromatin visualization. And third, we have the centromere position, which is important in mitosis and meiosis. So different chromosomal aberration examples. We have 4 right here. First, we have radiation that makes 2 breaks, and this results with non-homologous end joining and results in a simple aberration. Or radiation may make more than 2 breaks, which will result in, therefore, a complex aberration. So those 2 examples would be breakage and reunion. The third example would be recombinational misrepair by the DNA repair mechanism. So there's a radiation which makes a break in the chromosome. The DNA repair mechanism tries to fix the mistake, but actually makes a mistake in trying to fix the mistake, so we have an aberration in the chromosome. And lastly, the exchange theory, where radiation makes 2 lesions in the chromosome and reciprocal exchange occurs. Of course, this is also an aberration. Now we're going to show you some examples of karyotypes that show genetic disorders. So first, here we have XXY. As you can see in the karyotype, there's an extra X chromosome. And this person is actually genetically male, but they have an extra X chromosome. They present usually as infertile people. And again, this is very obvious in the karyotype. Next is Down syndrome, trisomy 21. So in trisomy 21, this person has a partial extra copy of chromosome 21. And usually these people present with characteristic facial features and intellectual disability. Finally, this is an example of another karyotype that would show a genetic disorder, Turner syndrome, XO. So this is a genetically female person, but she's completely missing one of her X chromosomes. So one of her X chromosomes is completely missing. These people present also with short stature, webbed neck, a low set of ears, and they're also usually infertile. So I hope this presentation was able to help you to understand some of the karyotype and why it's important, and a little bit about what a karyotype will show and how to make a karyotype. Thank you.